Bom dia. E aí, como é que você tá?
Thank you. More reading. Yes, yes. Good. Thank you. It's for me. Yes.
Ano que vem ele vai.
Tchau, tchau, tchau. Tudo. Fala, meu amigo. Armadilha do Yosmar. Coisa linda, você. Filhão, segura pra mim, senão eu vou enfiar no bolso. Boa. Ops. Boa tarde, pessoal. Eu queria agradecer muito a vinda de vocês aqui para assistir a palestra do professor Kinan Dandi. Essa é a última parte da visita deles aqui a Maringá, a última atividade nossa, é a palestra do professor Kinan. Obrigado por terem atendido ao nosso chamado e terem vindo assistir. Acho que é uma coisa que tem muito a ver com o trabalho que a gente faz aqui. Desculpa. O professor Kinan Dandi, ele é formado em Química na Universidade de Wheaton, com bacharel. O doutorado dele foi na Universidade de Michigan, também com Química. E ele fez um pós-doutorado Furman University em nanotecnologia. Em 2000 ele ingressou na Universidade de Illinois, em Springfield, e desde 2006 eles têm desenvolvido esse projeto de restauração da planície de inundação do Rio Illinois. Kinan, Dr. Dandi, thank you very much for coming to give us this lecture. It's a pleasure to have you here. And I guess that's all.
É um prazer conhecer vocês. Desculpe, eu só falo pouco português, so I will switch to English. But it has been a wonderful time for us to be here the past 3 weeks, to meet with you, to tour your labs here at NUPELIA. We also spent last week at Puerto Rico, and before that we got to tour Itaipu and the Cataratas Iguazu. So it's been a wonderful visit, and I'm hopeful that this is the continuation, actually, of a collaboration between UIS and UEM. Next week, some of you will be coming, and we will have the pleasure of hosting you to tour the Illinois River. And that's what I'm going to talk about this afternoon, the Illinois River. As Dr. Velio said, I'm a chemist, so I'm going to focus on the chemistry. I know most of you are biologists, so hold your biology questions to the end. But actually, if you have any questions anytime, feel free, raise your hand and during the presentation. And fortunately, your rector gave me this presente hoje, the laser pointer, so I can do this presentation.
I'm jealous.
So to start, I want to thank all of the students and faculty who have worked with me over the years on these projects I'm going to talk about. And primarily, right here, Professor Mike Lemke, whom many of you know, he's been here a few times. He is the one who dragged me out of the lab and got me in a boat on the water and started this journey of research for me in 2006. And there are many students have worked with us over the years. We've also worked with the Nature Conservancy, faculty and students from our sister campus. The larger, older campus has the DNA technology that we use. And we've also worked with biologists with the Illinois Natural History Survey. These are the people actually doing fish and waterfowl research that I won't be talking about today. And of course, our collaboration with UEM and NUPELIA. So just background for a general audience, why restoration and conservation is important for floodplains. So the theme of our summer exchange is conservation and restoration on the Illinois River and the Paraná River, comparing two great rivers in the Americas. And in particular, we're interested in conservation and restoration of the floodplain for these large rivers. This is important because this is the aquatic terrestrial transition zone where there are many different types of habitats, where you have lakes on the floodplain, wet marshes, wet meadows. Above the bluff, you have forests and prairies, or cerrado here, and natural levees, and of course the main river channel. So all of this different types of habitat provides different niches for multiple organisms and biodiversity. Another aspect of the floodplain is the flood pulse. This is data from the Illinois River floodplain comparing nitrate concentrations in the river, in the yellow dots, to the backwater, the green triangles. And during a flood event, the river waters mixing with the backwaters, and so all the concentrations are all over the place. But then after a few days, these green points go down, the yellow points continue to go up. So there's a separation of concentrations for the nitrate from the main river channel to the backwater channel. And so this shows the importance of the flood pulse in terms of transferring these nutrients. So flooding is a necessary event for the chemistry, and of course you guys already know about how important it is for biology. Now, over time in the Illinois River, the natural flood pulse has been disturbed by human activity. And this image here shows before major human interaction with the Illinois River floodplain. You have natural levees and you have different types of backwaters, which provide habitat for all types of organisms. Then here's from the 1960s, after human intervention, we have the channel's been dredged in the river, levees have been made, the land has been drained for agriculture, the water level on the Illinois River is higher than it was before, about 1.5 meters on average, and also has more sediment and more pollutants from Chicago. And this then causes this backwater lake that was still intact to get filled up with silt and also degrades that habitat. So this is what's happened over 100 years in the Illinois River floodplain. And I want to show you a couple case studies of restoration efforts. Here is the Illinois River basin running from Chicago down to St. Louis. And this restoration project site is right here called Emiquon near Havana, and it's owned by the Nature Conservancy. So this shows you in green is the land owned by the Nature Conservancy. There are other natural lands or conservation units here in the area owned by, this is federal, U.S. Fish and Wildlife. This is State Department of Natural Resources. And you can see the river is right here. So what has been done at this site? First, let's review some history of the site. So 1673 was the first time a European came to the Illinois River Valley, Franciscan monk Pierre Marquette and Joliet, and they saw Emiquon and they said, "We have seen nothing like this river we enter for its fertility." So this natural floodplain that was intact provided lots of habitat for animals and plants. And here we have in 1903 an actual picture on the site of Charles Cofoid, who was a scientist with the Illinois Natural History Survey started by Stephen Forbes. And he's in one of the sloughs with Thompson Lake measuring phytoplankton and things like that we like to measure. So that was 1903. Then in 1919, the local newspaper reported how dredges are building up a levee in this lake to drain it for agriculture. So that's the way it was from 1919 on. Here's a picture in 2003. This farmland here used to be the lake, and it was one of the largest farms in Illinois. The Nature Conservancy bought the land in order to conserve it for restoration. So here we see in 2007, they stopped draining the land. They turned off the pumps that were being used to drain the land, and so just the rain came and started to refill the basin. So by 2008, here's Thompson Lake again, here's Flag Lake again. So after 80 years, the floodplain is coming back. And that's what we've been studying is this process of restoration. How does that occur when you take an agricultural field and turn it back into the lake? And here just shows a time sequence of the water filling the basin. So that was that picture in 2008. But then by 2009, the water continued to fill the basin, and Thompson Lake and Flag Lake actually merged, and that's the way it is today. So there's about 3,000 acres of lake. Sorry for the non-metric system, but that's okay. So that's one restoration effort on the Illinois River floodplain by the Nature Conservancy. Another restoration effort also by the Nature Conservancy is at Merwin Preserve, where they started that in 1997, so almost 10 years ahead of Amequon. And so we have data comparing a more established restoration site to a brand new restoration site. And we've been monitoring both of these sites. It's a smaller site, but again, it's right on the Illinois River. It's unconnected to the river. So the next thing I'm going to talk about is the importance of the connection. And we did a study to compare what's the aquatic chemistry and microbiology for lakes, floodplain lakes that are connected to the river or periodically connected by the natural flood pulse versus hydrologically unconnected lakes. And our hypothesis was that the sites connected to the river would be more similar to each other than those lakes that are unconnected. And I'll show you some, here's where the sites are here. So this is what Amequon we've been talking about. Down here at the end of the LaGrange Reach is the Merwin Preserve, which is the other restoration site. Up here is another unconnected lake called Spring Lake. And here's where we sampled the river at Havana. This is Matanzas Lake, Chain Lake, and Crane Lake. These three lakes are connected to the river. And the 3 years we sampled was in 2006, 2007, and 2008. And it just happened, which is serendipitous, that we had 3 very different climates those years. So in 2006, it was a drought year. You can see the river never reached flood stage. In 2007, the blue line, where there was a spring flood, a summer drawdown, and then a small flood in the fall. So this is what the Illinois River is supposed to do. It turns out with climate change and all the man-made changes to the river, this is actually a rare occurrence. And then in 2008, we had floods, lots of flooding occurring, which actually happens more and more frequently in Illinois. It's actually, I think, flooding now while we're here. So we did see differences between the connected lakes and unconnected lakes, and we did see that the connected lakes were more similar to each other and more similar to the river. So for example, turbidity, you see in all 3 years, the turbidity is higher for the connected lakes than for the unconnected lakes. And also nutrient levels. So phosphorus is a limiting nutrient in these systems. You see that connected lakes have more phosphorus and are more similar to the river than unconnected lakes. So this connection is important for providing those nutrients and therefore helping with growth of all the biologicals. Okay. One aspect of biology that actually I don't know that much about, but Mike Lemke, my collaborator, does this research in terms of bacteria communities. And the technique he uses is called ARISA, Automated Ribosomal Intergenic Spacer Analysis. And we're looking at the RNA in the bacterium and looking at this space between the genes that the primers are for the genes. So this space is going to vary depending on your species. So you have these different bacteria, and so you get you can separate the species based on this spacing. And this doesn't actually tell you the identity of the species, and actually it could be that you have 2 species that happen to have the same or very similar spacing. So you can't tell exactly how many different species you have, but you get a snapshot of the diversity of the species and some quantification information. So you can get bacterial community composition. And when we compare the bacterial composition, community composition of the connected lakes to the river, to the unconnected lakes, we see that there's actually some separation here. This is averaging of all 3 years that we sampled, all 3 summers. You see that on average, the connected lakes are closer in bacterial community composition to the river than the unconnected lakes. So again, this hypothesis that the connection makes things more similar, it makes sense, right? The river is inoculating, if you would, the lakes with bacteria. Okay, so that was a study we did a while ago. Since then, the connections that we were talking about have changed. So in 2013, there was a flood, another flood on the Illinois River. This time it was the highest of the 150 years of record keeping. And this caused a change in the connection. Our Emiquon Preserve, Thompson Lake, used to be unconnected to the river, became connected. Merwin Preserve, Long Lake at Merwin Preserve, was unconnected and became connected. So what happened to these restored floodplain lakes when the river reconnected itself? Nature did this experiment, and we were fortunate to be there and get the data. And it was accepted very recently for publication in I've heard this is like one of the most important journals in the world, right? So I'm going to talk about the results from this publication. So here you see in the red, this is the height of the river. During the flood, you can see it broke the previous record from 1943. And at these points here is where the water from the river is overtopping the levee and entering Emiquon. And so you can see in green the depth of the lake. The lake height is increasing as it's getting filled by water from the river. Now this is a photo showing you the water overtopping the levee. And I don't even think you can see the Illinois River here. This is backwater from the river. This is Emiquon here. So the levee remains intact, but as water comes in from the river. This shows some of the chemistry and hydrology here. Again, here's the flood event. Here's the lake level going up a little bit. During the flood, there's a spike of nitrate. So unlike what you have here in the Paraná, we have, in a sense, the opposite problem. So we don't have dams like this, we have dams like this, the levees. And what that causes, instead of depleting the nutrients, it adds nutrients. So we have too much nutrients in our river, and so when we had the flood event, it added nutrients to our floodplain lake. But you can see that the nitrate quickly was consumed by the system, and then there was a peak in the phosphate later on in the summer. What happened to the bacteria during this event? So this is again a plot of the bacterial community composition and flood. Damn Google Drive. Okay. there is a data point here. Okay. Flood and then after the flood. Basically what you see is not much change. I mean, you could imagine that there's something happening maybe right here, a little bit of a shift. But as we saw in the case of the chemistry, whatever change, whatever inoculation came from the river was quickly disseminated by the floodplain lake. Okay, at Merwin Preserve, the other previously unconnected, restored site, the story is different. This is a cross-sectional view of the levee, which you can see we're now in water. There's no levee here anymore. The river blew out the levee. Here's the river here, here's the long lake, and you can see it's now connected. So this was right after the flood, or during the flood event. A year later, there's still the notch here in the levee. This is the top view of this same site. And here's the river, and the lake now has drawn down. So that's what's happened at Merwin Preserve. We now have a connection with the lake. Every time the river floods, there's going to be river water into the lake. And what happened hydrologically, you see here's the flood event. we started sampling in the middle of the flood, and as the waters receded from the flood, the lake drew down until it was less than 0.6 meters here, at which point we have not just a shallow lake, but a very shallow lake. And so turbidity goes way up, as well as chlorophyll. With all these nutrients from the river, we have algal bloom. What happens to the community? Here you can see a clear shift between what the lake, when it's not connected, not flooded by the river, and what the lake bacterial community looks like when it is connected during a flood event, which happened actually multiple times throughout the year. Throughout that year, there was even a flood in January under the ice. So flooding is important for these systems, and there's some positive outcomes of this flooding event. This is a measure of denitrification rate. So it's amount of nitrate being converted to nitrogen gas, which removes then this excess nutrient from the system. It goes into the atmosphere and goes away. So this is an important bacterial service, if you would. And you can see that following the flood event, denitrification of the soil bacteria increased dramatically. And if we compare the lake, like in the middle of the lake, the sediment, as opposed to at the edge of the lake where it flooded the prairie soil, we see that the flooded soil actually does better at denitrification than just regular lake sediment. Now when we compare that with what happened at Merwin, where the levee was broken and so we now have open connection with the river, we see again an increase in denitrification rate for the flooded soil, as well as after the flood, there continues to be an increased denitrification rate. So the flood event actually helps the denitrifiers, probably because it's adding another nutrient we heard about yesterday, carbon, to the system in the form of detritus from, say, the flood event. Okay, so we saw how if connections make a difference and how important the connection is. What about the type of connection? So there's two models you could think of for how a lake, a floodplain lake, is connected to the river. One would be where there's a sequence of events where river water goes into the lake, is processed by the lake, and then comes out of the lake. So you could imagine, in chemistry we would call that batch processing. And so to visualize that, you have the human impacted water, it comes into the lake, gets processed, filtered if you would, and then the output would be water with improved water quality, like less nitrate, for example. Another model would be where the water comes in one side into this wetland and then comes out the other side. And this is actually another site on the Illinois River that we're going to visit in a couple weeks. And this is your classic wetland where it acts like the kidneys, right? The water from the river gets spread out, processed, and then exits the wetland cleaner, just like your blood in the kidneys. And so you could call this a continuous processing model. And the processing we're talking about would be biotic, like the bacteria denitrifiers, or it could be abiotic, or some chemical processes, precipitation, sedimentation, other things like that, pH changes. So these are two different types of systems, which would depend on the nature of the connection between the floodplain and the river. And this is the next step at Emiquon, which is an actual connection with the river. So before we saw Emiquon got connected when the river flooded, now the Nature Conservancy has built a control structure. Here's the levee at Emiquon. So now they can open and close the connection with the river when they want, depending on what they want to do. And so this could allow for, say, a batch processing scenario where they could open the gates, let the river water in, close the gates, let it be processed, and then open the gates to let the river water out. And they installed this last summer, and I have no data yet on whether this does what perhaps this model could do. And as I understand it, they are going to be sending out a call for proposals. So if you have ideas on how perhaps this structure could be used and what science could be used, there are multiple bays here or ports here where you could put instrumentation or cameras to track fish movements and things like that. So there's a lot of opportunities there, and hopefully next time we'll have some data to share from there. So to summarize, I talked about this ATTZ concept, how important the floodplain and the river connection is, how we have nutrient inputs as well as inoculation of life forms, bacteria, perhaps zooplankton, fish, and I know a little bit about what happened to the fish during the flood, but if you have questions about that, I'll have to ask someone for a lifeline. But type of connection matters. Partial connection, we saw at Emiquon, it had input from the river, but the system quickly returned to the way it was before the flooding. Whereas open connection, we had a very dramatic change in the river water quality, or the water quality at Merwin Preserve due to the connection with the river. And one or two or more connections you could design different scenarios for processing these waters. And with the ultimate goal would be to restore perhaps the Illinois River, right? This is the theme of our session here, conservation and restoration. So the Illinois River has been impacted in many ways over the past 100 years by humans. So how can we help to improve the river, restore some of the services? And thank you for your attention. These are our funding agencies. So.
Okay, questions?
Go ahead, Amber.
I have 3, so you
Oh my gosh. I don't think I need that.
Yeah, because they got recording.
They're broadcasting.
Okay.
you want to you just open it up.
10 minutes.
10 minutes to British Amber. Now you are a pop star.
To open the un momento.
I don't know how to open
There's that side.
Good. Can I ask now? Okay. With the theme of like conservation and restoration being to bring it back to as natural as possible, if the lake isn't naturally connected, why build a building that connects it to the river?
Yeah, so that's a good question. The idea of having a controlled connection is because we saw what happens when you have an open connection at Merwin Preserve, and there are other examples up and down the Illinois River Valley of where people tried to restore the wetland, the floodplain, and the open connection, it degraded the system so much that it can't be used for habitat for migrating waterfowl and rare fish and things like that you want. So that's why the Nature Conservancy has a control structure. They don't want it just wide open. And part of the reason is because the Illinois River is so degraded that the idea of restoring the floodplain back to 1903, pre-1919, I mean, that's a fiction. No one can do that because the river is not, the river itself is not pre-1919. The river has been impacted by Chicago. There's a canal that connects the Chicago River to the Illinois River. Lake Michigan water comes down, as well as treated sewage from the city of Chicago, as well as the millions of people up and down there. So, you know, that's a good question, and that's, I think, one of the philosophical discussions about restoration is, and we've been talking about that all this time, is conservation, restoration. What is conservation? What is restoration? What does it mean to restore a system when the world's changed? So, yes, Amber.
First question, just for clarification.
That's the second question.
Matanza Lake, is that connected, unconnected, or temporarily connected?
So Matanzas Lake is connected to the Illinois River, yeah, and we're gonna go there next week.
Okay. Second question. You talked about denitrification, but you didn't really touch on dephosphorylation, but phosphates impact freshwater eutrophication, while nitrogen and nitrates impact marine eutrophication. So is there any data for dephosphorylation?
Yeah, so phosphorus is actually more of a limiting nutrient in these systems usually than nitrogen. And there is excess phosphorus in the river as well. I don't know, is there a faster way to do this? Anyway, but what my data showed is that backwater lakes have less phosphorus. So there is, I don't think dephosphorylation is the term, but there is processing of the phosphorus. It goes into the plant matter, for example, and then the ducks and fish eat it and then they leave. So phosphorus can be removed. Phosphorus also can be removed by sedimentation and precipitation. So.
I forgot my third one. So.
Okay. Oh, good. But we can come back when you remember.
Everybody else with the torch.
Okay. Professor Rocha.
Professor Keenan, thank you very much for your talk. I think it's not a question, but just an addition to the talk, because when you talk about the dams, here in the floodplain, we have a cut in the river, as you told, and in your case, we have lateral levees. So in this case, when you see the dynamic of the floodplain lakes, we can see that the nitrogen is really dynamic before and after the flood. But I tried to do some analysis on the phosphorus. I didn't find the pattern in nitrogen here, but in phosphorus. When we separate the lakes between connected and unconnected, then we can see that the connected lakes have no dynamics of phosphorus a long time, but the connected lakes has. If you look the phosphorus before the flood, then you can see that the phosphorus is high, but during the flood, they become lower, and then start again to become higher after the flood. So it's a really different dynamic from your floodplain to our floodplain.
Right.
Yeah, that's quite interesting, because as you told, the phosphorus can be gone with plants and other stuff like iron available in the water, then can sedimentate phosphorus in the sediment. But here, phosphorus remains in the water, so it's really different dynamic. Maybe we can try to compare different kinds of dams and the effects on nutrient dynamics in the shallow lakes.
That sounds great. Let's just get together and write a paper.
Probably. So, but that's to show you a different perspective on the nitrogen and phosphorus dynamic that we have in our floodplain. So it's really interesting.
Very, very interesting.
I don't know about the bacteria and other stuff, but I tried to analyze the nitrogen and phosphorus, and we have this different.
Yeah, great. Thanks. Yeah, good.
More questions? I have one. Actually, we have talked about it this last weeks, right? But the question is the new gates that you can control, the amount and the time of water get in the Thompson Lake, how the possibility scientists work on this experiment, I would say, right? Open the gates and for a long time, for few times, for a lot of water, less water, and all other kinds of experiment, because you know our 30 years working here, we know that the intensity and also the duration of the pulse, it's very, very important for the community, right? What the possibility to work on this possibility of experiment about duration and intensity of floods?
Yeah, that's a great question, and I'm not in charge of that, so I don't really have an answer, except the Nature Conservancy owns the site, but they are very interested in science. And so next week, when we meet Doug Blodgett, who's director of this program, you can ask him that question then. But when they started Emaquan, they assembled a team of 40 scientists who met periodically to design how was the site going to be managed. What are the what they called key ecological attributes that should be measured to determine is the restoration successful. And so scientists have been involved all along. Currently, as I understand it, the opening or closing the gates is controlled by the fact that there's too much water at Emaquan. I showed you that picture in 2009 where the lakes have merged and the water has continued to rise, and it's now damaging the levees around Emaquan, which includes a state highway. So the state will sue the Nature Conservancy if they damage the highway. So the Nature Conservancy is very interested in draining Emaquan right now, which means not opening for flood. It's opening for water exiting, not incoming. So that's, while they're interested in science, they also have these pragmatic issues for managing the land. Yeah.
More questions? No? So, Dr. Danji, thank you very much for your lectures, for your talk. It was really great, and I would like to say that was really great to have you and your students here with us for this 3 weeks, right? Thank you very much for coming, and I hope you enjoy and work a lot from Chicago to St. Louis, right?
Right.
From Chicago to St. Louis. Gente, muito obrigado por terem vindo, por terem participado, por terem contribuído dessa nossa primeira experiência, né, aqui de intercâmbio com o pessoal lá dos Estados Unidos. E espero que tenhamos outras oportunidades ano que vem. E aí vocês estão todos convidados a participar também. E como o Dr. Danji said, if you have ideas to work with this with this question, like Dr. Roger said, we're very welcome to discuss with us. Okay? Thank you very much.
Okay.
Muito obrigado.
Muito obrigado.